The pteridophytes include hostels and fawns. Pteridophytes are used for medicinal purposes and as soil menders. They also frequently grow on parameters. Evolutionary, they are first terrestrial plant to possess vascular tissue, xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phases in life cycle is gametophytic plant body. Gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte, which is differentiated into true roots, stem, and leaves. These organs possess well differentiated vascular tissues. The leaves in pteridophyta are small microphyll as in Selaginella or large microphyll as in fern. The sporophyte bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structure called strobili or cones. Selaginella equisetum. The sporangia produce spore by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular. Free living. Most free living. Mostly photosynthetic thyroid gametophyte called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the speed of living pteridophytes is limited and restricted to a narrow geographical regions. The gametophyte wear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer for antherozoids. The male gamete released from antheridia to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produce a multicellular, differentiated sporophyte, which is dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are similar kind. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro, large, micro, small spores, are known as heterosporous. The megaspores and microspores germinate and give rise to the female and male gametophytes, respectively. The female gametophyte in these plants are retained on the parent sporophytes for variable periods. The development of zygote into young embryos take place within the female gametophytes. This event is a precursor to seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, Lycopsida, Sphenopsida, Pteropsida. Gymnosperm. The gymnosperms are plants in which the ovules are not enclosed by any oval wall and remain exposed both before and after fertilization. The seeds that develop post-fertilization are not covered, that is, are naked. Gymnosperms include medium-sized trees and tall trees and shrubs. One of the gymnosperms are giant redwood trees. Sequoia is one of the tallest tree species. The roots are generally taproots, roots in some genera have fungal association in the form of mycorrhiza, pinus, while in some other cycles, small specialized roots called caraloid roots are associated with nitrogen fixation cyanobacteria. The stem are unbranched cycles and branched pinus and catarrhus. The leaves may be simple or compound. In cycas, pinnate leaves persist for a few years. The leaves in gymnosperm are well adapted to withstand extreme temperatures, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and second stomata also help to reduce water loss. Some are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophyll, which are arranged spirally along an axis to form legs or compact strobili or cones. The strobili bearing microsporophylls and macro microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called pollen grain. The development of pollen grain takes place within the microsporangia. The cone-bearing megasporophyll with the ovule or megasporangia are called macrosporangiate or female strobili. The male or female cone or strobili may be borne on the same trees, pinus. However, in cycas, male cone and megasporophyll are borne on different trees. The megaspore mother cell is differentiated from one of the cell of the nucellus. The nucellus is protected by the envelopes and the composite structure is called an ovule. The ovule are borne on megasporophyll which may be clustered to form the female cones. The megaspore mother cell divided meiotically to form 4 megaspores. One of the megaspore enclosed with megasporangium develop into a multicellular female gametophyte that bear 2 or more archegonia or female sex organs. The multicellular female gametes is also retained within megasporangium. Unlike bryophytes and pteridophytes, in gymnosperm, the male and female gametophytes do not have an independent free-living existence. They remain with the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovule borne on megasporophyll. The pollen team carrying the male gametes grow towards archegonia in the ovule and discharge their contact near the mouth of archegonia. Following fertilization, zygote develops into an embryo and the ovule into seeds. These seeds are not covered. Angiosperms. Unlike the gymnosperm where the ovule are naked, in angiosperm of flowering plants, the pollen grain and ovule developed in a specialized structure called flower. In angiosperm, the seed are enclosed in fruits. The angiosperm are an exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest wolfia to tall trees, eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicines, and several other commercial important products. They are divided into two classes, dicotyledons and monocotyledon. The dicots are characterized by seed having two cotyledons, reticulate venation in leaves, and tetramerous and pentamerous flower that is having four or five members in each floral wall. The monocotyledons, on the other hand, are characterized by single cotyledon seed, parallel venation in leaves, and trimerous flowers having three members in each floral wall. The male sex organ in flower is a stamen. Each stamen consists of a slender filament with an anther at the tip. Within the anther, the pollen mother cells divide by meiosis to produce microspore which mature into pollen grains. The female sex organ in flower is a pistil. Pistil consists of a swollen ovary at its base. The long cylinder style and stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore cell that undergo meiosis to form four haploid megaspores. Three of them degenerate and one divide to form the embryo sac. Each embryo sac has a three-cell egg apparatus, one egg cell and two synergids, three antipodal cells and two polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain after dispersal from the anther and carried by wind or various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grain germinate on the stigma and resulting pollen tube grow through the tissues of stigma and still reaches the ovule. The pollen tube enter the embryo sac where two male gametes are discharged. One of the male gamete fuse with egg cell syngamy to form zygote. The other male gamete fuse with the diploid secondary nucleus to produce the polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain after dispersal from the anther are carried by wind various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grain germinate on stigma and the resulting pollen tube grow through the tissues of stigma and style and reach the ovule. The pollen tube enter the embryo sac where two male gametes are discharged. One of the male gamete fused with egg cells syngamy to form zygote. The other male gametes fuse with the diploid secondary nucleus to produce the triploid primary endosperm nucleus PEN. Because of the occurrence of two fusions, that is syngamy and triple fusion. This event termed as double fertilization, an event unique to angiosperm. The zygote develops into an embryo with one or two cotyledons, and the PEN develops into endosperm, which provide nourishment to the developing embryo. The synergids and antipodal degenerated after fertilization. During these events, the ovule develop into seeds and the ovaries develop into fruits. The life cycle of an angiosperm is shown in figure 3.6. Plant life cycle and alternation of generation. In plant, both haploid and diploid cells can divided by mitosis. The ability lead to the formation of different plant bodies, haploid and diploid. The haploid plant body produce gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divided by mitosis to produce diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexual reproducing plant, there is an alteration of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in the following patterns. Number 1. Sporophytic generation is represented only by the one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote result in the formation of haploid spores. The haploid spores divided mitotically and form the gametophytes. The dominant photosynthetic phase in such plant is the free-living gametophytes. This kind of life cycle termed as haplontic. Many algae such as volvox, Spirogera and some species of Chlamydomona represent this pattern. Number 2. On the other extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase represented by single to few-celled haploid gametophyte. This kind of life cycle termed as diplontic. An algae fucus species represent this pattern. In addition, all seed-bearing plants, that is gymnosperm and angiosperm, follow this pattern with some variations, wherein the gametophytic phase is few to multi-celled. Bryophytes and pteridophytes interestingly exhibit an intermediate condition, haplodiplontic. Both phases are multicellular, however, they differ in their dominant phases. A dominant independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte and it alternates with the short-living multicellular sporophyte totally or partially developed and dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. Diploid sporophytes is represented by a dominant independent photosynthetic vascular plant body. It alternates with multicellular sporophytic autotrophic independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most algal genera are haplontic,